Hi, everybody. I'm Caroline. I'm a postdoc at the University of Virginia, and I'm going to talk to you today about interactions between avoidance and resistance to parasites. So many of us, when we think about defenses against parasites, we think about immune systems, we think about physiological defenses. But, I think I need to get back into this. Okay, but studies of invertebrates and mammals and these really amazing lemurs that I studied for my PhD and recent human events in our society have shown us that behavior is a really important defense against parasites that is really ubiquitous among animals, and that's specifically avoidance behavior. So when a host is around a parasite, it can avoid that parasite by not contacting it. And then after it makes contact, it can use post-contact defenses to resist infection. And so avoidance and resistance both have a shared outcome. They both reduce infection. And because of that shared outcome, they may not vary independently of each other. And so I'm going to tell you today about two alternative hypotheses that for how avoidance and resistance might co-vary, and then an experiment that I did to test those hypotheses, and end by telling you a little bit about some ongoing future directions. The dominant hypothesis in the literature is that avoidance and resistance should co-vary negatively, or some people call this a trade-off, because they both entail costs, but they have a shared or common function. And so a study of house finches showed that individuals that had lower immune function were more likely or less likely to less likely to avoid their sick conspecifics. And so they found the evidence supporting this negative covariance. And it raised a lot of questions about whether there is a genetic basis for this covariance or whether these traits actually functionally reduce infection independently. Alternatively, avoidance and resistance might co-vary positively, especially if they involve shared pathways or mechanisms. A study of this flowering plant, Silene latifolia, showed that families that flower later in the season, and thereby phenologically avoiding the spores of this fungal parasite, were also families that were more resistant when they were directly inoculated with the fungus. And so this study was really nice because it showed that there was a genetic basis for this positive covariance, and by directly inoculating the hosts, they were able to isolate the effects of resistance independently of avoidance, and so they're separating these two defenses. So working with animals, it's a little bit harder to directly inoculate, right? But I wanted to try to find a host system where we could look at behavior and physiological forms of defense separately. And the host that I work in is Caenorhabditis elegans, the model nematode. You can see it here with the parasite that I study, Serratia marcescens. And I studied 12 genetically diverse strains of C. elegans to capture a broad range of genetic variation. These are strains that are publicly available via the Caenorhabditis Natural Diversity Resource. And I focused on two distinct strains of Serratia, DB10, and it's a little hard probably to read that red text, but that second strain is SM2170. So in this host parasite system, I, along with this recent graduate of UVA, did a a lawn leaving assay to quantify behavioral avoidance. And so what we did was to add 20 individual hosts to the center of a standardized size and shape patch of either DB10 or SM2170. That patch is called a lawn. And so then 18 hours later, we counted how many individual hosts stayed on that lawn and how many left to the empty agar surrounding it. And then we quantify avoidance as the proportion of hosts that are off of the lawn over the total hosts that were counted. So I'm going to show you the variation in that trait for each of the two parasite strains, DB10 and SM2170. Along the x-axis are the 12 C. elegans strains, and the y-axis is the proportion of hosts that are avoiding or leaving that lawn. And I'll just set your expectations that when we do the controls in a non-parasitic strain of bacteria, we see really low levels of avoidance. The worms pretty much stay on lawns of bacteria that are not pathogenic, and that's because they're bacteriivores, so that is their food. They like bacteria, except if they're parasites. And so you can see when we look at the behavioral responses to the two parasite strains, first of all, we have higher avoidance of DB10 overall than of SM2170. But within each of those strains, we see a high degree of variation across C. elegans strains. And we also see a significant strain-by-strain interaction, indicating that these responses are really strain-specific and depend which host and which parasite strain you have paired together. To quantify resistance, we really wanted to try to directly inoculate these hosts and measure resistance bypassing avoidance. And so this excellent undergraduate, Adiana Lockwood-Shabat, and I have put individual C. elegans worms into the wells of 96 well plates and added concentrated doses of the parasite. When the nematodes are in liquid, they become filter feeders. And so they essentially indiscriminately ingest the parasites that are in liquid with them. And then we can quantify resistance as the proportion of hosts surviving 48 hours later. And so, again, I'm going to show you C. elegans strain on the x-axis. You notice that the strains are numbered in a different order here. The number corresponds to the previous, but they're in a different order. And then the y-axis is the proportion of hosts surviving, where 1 is complete resistance to the parasite. And I'll just show you that we wanted to account for the fact that strains might vary in their performance in liquid at all. So we did a control where there was a non-parasitic strain of bacteria. And we did find that there was some variation in performance. This one strain did really poorly in liquid, and so we want to take that into account when we consider the effects of the parasites. When I add the parasites on here, you'll see again that overall survival or resistance is much lower in response to SM2170 than to DB10, but there's a huge range of variation, and again, we have significant strain-by-strain interactions here. So resistance is also highly specific. So now I have everything I need to test my main hypothesis: do avoidance and resistance covary? And so I'm going to plot resistance on the x-axis here, that's the proportion surviving, and on the y-axis it's the proportion leaving that lawn, that circular patch. I'm showing you for DB10 first that there is no relationship here. And I separate the two parasite strains because we saw that these are really specific interactions. So for DB10, we see no significant relationship between avoidance and resistance. And the same is essentially true for SM2170. We have a marginally negative relationship here. So there is maybe like some suggestive evidence for negative covariance, but remember that this is a strain that overall defenses are really low against. So, take that for what you will. So to summarize those results, we have a really specific strain-by-strain pattern of interaction here for both avoidance and resistance. This is fairly common to find for resistance, but I was really surprised that the interactions were so specific for avoidance. We tend to think of avoidance as a really general defense. If you think about behavioral defenses in other animals, it may not matter what the genotype of the parasite is, right? But for C. elegans and Serratia, it seems that this is a really specific interaction. And that means that the theory developed for resistance could actually be relevant for, predictions for avoidance evolution or coevolution. But overall, we see that there is not a consistent covariance across natural C. elegans strain for either strain of Serratia. And so, that's sort of like a null result, but it's kind of exciting because it means that we can separate these two defenses. They're not inherently linked in some way that makes them impossible to separate. And that also makes it possible to test a hypothesis that I think could actually affect the covariance of these two traits, which is what I'm sort of doing next. So for my future directions, rather than assuming that there are costs or that there's a mechanistic interaction between these two defenses, I'm really interested in how the sequence of avoidance and resistance defenses influences their covariance. And so if we think about infection as, proceeding, into the host body and establishing itself, our defenses can be thought of as filters that have sort of wider or narrower openings. And avoidance is a filter that occurs before resistance. And so if all of the parasites that make it to the resistance filter are first blocked by the avoidance filter, perhaps the nature, the magnitude, the intensity of that avoidance defense is going to determine how parasites actually select on resistance. And so I'm actually currently doing an experimental evolution project where I'm evolving elevated and diminished avoidance behavior independently of resistance in C. elegans, and then seeing how those higher and lower avoidance defenses affect downstream resistance evolution. So at a future meeting, hopefully I'll be able to tell you more. And with that, I'd just like to thank my postdoc advisors, members of the Gibson Lab, the excellent team of undergraduates who have contributed to this project, especially Adiana and Andreas, and I would be happy to take your questions. Yeah, I don't think we're going to have dings, so I'll just try to watch the time. Yeah.
So I was wondering, have you ever done this with like a male bridge strain?
There is variation in males in those strains, but no, not a male enriched strain. Is there a reason?
No, I just think it would be cool if there was some sort of sex difference in terms of avoidance, because males do more of the mate-seeking behavior, so I can see it and if there is an effect, maybe step back and then do it in an obligately outcrossing species as well, right?
Yeah.
I don't know, it would be cool.
Yeah, totally. My experimental evolution is actually with an obligately outcrossing, genetically diverse population. So yeah, because we want to have as much genetic diversity and opportunities for recombination as possible. So actually, we will have some data for an outcrossing population. Yeah. Yes.
Hi, by any chance, did you measure for the avoidance essays, the feeding rates, do they differ in that? If they're trying to avoid it, does it affect the feeding rate, or are you considering measuring feeding rate experimental issues that reduce feeding rate by default?
Yeah, good. The question was whether I'm measuring feeding rate. We have not, but I know it's something that is done in some studies, and it would be potentially another behavioral assay to do with a little bit more fine scale, maybe resolution. Yeah, I haven't done that yet. Okay, I think I'll hand off to whoever's next.
Thank you all for coming to my talk. My name's Drew. I'm working at Reed College as a visiting assistant professor. I'll be stepping into a visiting scholar position after this year. So I've been able to have my own little lab for 2 years, and it's been a lot of fun. So a lot of the work you're seeing is done with undergrads at Reed College. And basically, before I get started, I want to talk about this term cross-sexual transfer. This is an idea proposed in 2003 by West Eberhard. And I'll get to this picture in a second. And it's starting to get renewed interest within the field of evolutionary biology. Myself and some other researchers have been putting this idea out there as something we should be testing more, as it could explain a lot of the diversity we see in sex-biased traits. So the classic example that we found is this example of the white-shouldered fairy wrens. We have an ancestral morphism up there at the top, the alboscapulatus complex. Wow. And up there you have the male who's got these dark black colors and the white epaulette, and the female is very drab, so we've got this classic sexual heteromorphism. And then the more derived populations, you actually have the females taking on the male characteristics. They have that white epaulette, they've got the darker coloration. And West Eberhard proposed that this is probably due to some hormonal regulation effects that are happening, such that females are accessing the male hormonal regulation pathway to access these male traits. And this is an idea that I've been playing with and trying to look at more detail, but at the first level, we've been looking at global hormones. So the animal that I'm doing this in is Gelitochromis transcriptus, and just to give you some life history on them, they're these biparental cichlids. They're really cool. They pair up, they get in the nest, they lay eggs, they do teamwork to take care of them. And we think this evolved from male territorial behavior. So basically the males were holding territories and just kind of guarding general areas, and that eventually evolved into the two sexes cooperating to take care of the offspring. And the way Gelidochromys transcriptus prefers to work is they usually like to get in male larger pairs in the wild, so the female's small, the male's large, and then they take on these defined roles in parental care of the nest. But what we've actually found in the lab is we can create a flexible partner dynamic where the different sexes are taking on different traits based on their relative size to their partner. So just to show you, because videos are always a lot more fun, on the left here that's playing, we have a large male, small female pair that we've set up. And you can see they're both in the nest. They're kind of communicating with each other, giving each other little nips, which we call approaches. So the males, they'll swim towards each other with their mouth open and either make contact or not. And it's just a way they interact with their partner. And they'll also do what we call shimmies, where they'll shake. It's usually done in the nest, and it's usually done by the smaller fish. But the larger fish usually spends more time circling the nest, kind of acting as a guardian for larger for intruders. And we've definitely found when we do intruder experiments, the larger fish is the one that drives them off. And then the smaller fish stays in the nest and might do some more egg care behaviors. Now this video on the right that I've started up here, this is actually a female larger, male smaller pairing that we've set up. And you can see right there, that's the female who darted out, and now she's going to patrol the outside of the nest. So she's taking on the guardian behavior, and the male's taking on the more caretaker behavior inside the nest. And what I should also point out is this is the exact same female in both situations. So we've totally gotten that female to switch her entire behavior by giving her a different male of a different size. So we wanted to make a couple predictions based on this cross-sexual transfer idea and the idea that hormones should be regulating. Is when females take on this guardian behavior, they should be using male bias signals and male bias signal pathways. And the corollary, males should be using female bias signals to undertake this caretaker behavior. And so what we did is we took 8 females, 8 males, and we exposed them to larger and smaller partners of the opposite sex. And we just watched what happened behaviorally. And then at the end of each round, we basically took their hormones and looked at what changes we saw. So pretty straightforward experiment. And so confirming what I was telling you, the guardian behavior is male bias. So you're going to see these plots a lot. They're going to turn into reaction norms, but they are box plots to start. And so we can see on the left is males. Males are going to be in green. Females are going to be in orange. And we basically have male larger, female smaller. That's the typical pair that they make in the wild. And then the next one I'll show you will be the opposite direction, female bigger, male smaller. And so as we can see, the males engage in more guardian behavior than females. And we switch out the roles, and it's totally size dependent in how the behavior works. So the larger fish does the behavior, as I was telling you before. We confirmed it here with ours. But what's really interesting is it's not so much that both sexes are changing the behavior. It's the female who's really ramping up her behavior to kind of compensate for the male not going down very much, is what we see. So the females are spending way more time out of the nest. They're doing way more approaches than their male counterparts are, and the males are only slightly decreasing those behaviors. So we said, all right, well, those should be regulated by male bias signals, so we predict that those should be androgenic. So we took a look at 2 androgens. We looked at 11-ketotestosterone and testosterone. And fishes, just to orient you, 11-ketotestosterone is the sex-biased androgen. Testosterone doesn't tend to be very sex-biased. In fact, in our fish, it's actually going to be female-biased, which is pretty interesting. So it's ketotestosterone is going to be the main sex-different driver. And so as we can see here, we have this little bit of difference before. But then, when we have them switch their roles, same effect. Females go way up, and not only does she go way up in 11-ketotestosterone, which is what we would expect, she also goes up in testosterone. So she's using these androgens, increasing her circulating levels to get to these male bias traits. So far, so good. So we take a look in the other direction. We look at caretaker behavior, and this is the shimmy behavior. And this one is so dependent on what their partner does that I had to create it as a difference, because if I showed you the raw data, it wouldn't look as clean. So the males just shimmy way less than their small female partners. They just really do not shake very much as their females do. And then when you reverse it, they shake a lot more. They give the shimmy a lot more, but they still don't quite reach that female level. So we actually have a sex bias that is still in place. So this is not really a size bias, it's a sex bias. But males are attempting to shimmy more to kind of catch up to that level. And then when we look at hormones that might be driving those, we took a look at progesterone and estradiol. And again, both of these are female biased, which, all right, we're on the right track so far. And in fact, I want to draw your attention to just how much difference there is in estradiol. I had to log transform the plot in estradiol just to get males and females to be visibly plotted at the same time. And what we find is there's not much change in females in estradiol or progesterone, but males actually decrease estradiol, which is the opposite of what we would expect as they go to the caretaking behavior. So we have all these nice little correlations that we can see visually, but we decided to actually go ahead and throw them into models and see if we could actually model this behavior. So in this version, we're just using one predictor, each hormone at a time, and seeing what happens. So 11-ketotestosterone on its own can predict the behavior of out of nest as well as testosterone. So both of these androgens can predict behavior out of the nest. And then we did more complex modeling where we actually tried to optimize the model by adding one, dropping one approaches. And it turns out the best model is 11-ketotestosterone plus estradiol. So having both of those in the model, you can explain the behavior out of nest pretty well. And what I think is happening, again, is 11-ketotestosterone is driving a lot of it, and estradiol is probably kind of showing that female-male differences in how much they change. Then with approach, again, 11-ketotestosterone is our main culprit. We see estradiol again, and that's because the males drop it when they go smaller. But then when we try to do the model, basically your approach behavior is almost not dependent on any hormone. It's dependent on what your partner's doing. So the more time your partner spends out of the nest, the more approaches you do. The more time your partner approaches you, you approach back. The more time your partner shimmies, you approach. So there's this very much a dynamic interaction that's happening with these fish. And then there's a negative correlation with progesterone, which I'll tell you about after I show you shimmies as to what I suspect is going on. So shimmy has a negative correlation with progesterone, which is not what we expected. And then when we did the model, we got all sorts of crazy stuff. As I told you, partner shimmy is the big one. The more your partner shakes, the more you shake back. There's that negative correlation with progesterone again. And then a sex size interaction, and that's again because the males are the ones doing a lot of the changing in that one. And I really have been struggling to figure out why there's that negative correlation with testosterone. But it's there. It makes the model marginally better by adding testosterone. So showing it there for you. Now what's happening with progesterone is these fish are together for 2 weeks because sometimes they like the partner you give them, sometimes they don't. And so we give them that full 2 weeks to make sure that they're happy, they've partnered up, and they're getting along, and they're not going to kill each other, which sometimes we have to break them up if you don't give them the right partner. They're like, I don't like that one. But sometimes you give them a partner, and they're just like, this is the best partner you could have given me, and they'll spawn within 1 day. And that means that they'll be fry in the nest when we go do the behavioral observations. And once the fry are there, they stop paying attention to each other, and they pay attention to the fry and start doing those behaviors. And when they have fry, their progesterone levels go way up. So this only happened in 4 of the 16 pairs that we looked at, so I can't really model fry as a thing to look at, but I can tell you that progesterone is a pretty good proxy for whether or not fry are present in the nest. So that's what's going on in these behaviors. So time to check and see if this cross-sexual transfer hormonal regulation prediction holds up. Do females engage in guardian behavior by using male bias signals? Yeah, we've got pretty good evidence that that's what's happening. The corollary, do males engage in caretaker behavior by using female bias signals? No, not a lot of strong evidence for that. But as anybody who studied hormones and we think about evolutionary pathways, there's a lot of space between global hormone level and the behavioral output that you see. In fact, I want to study all these steps in between, and so this is something that I'm planning on getting after. And the great news is all those fish that we finished with, we were able to collect them and dissect out the brains, and we've taken brain punches of 3 regions of the brain, 2 associated with social behavior, the POA and the VD, and then 1 associated with visual behavior, which is the optic tectum, which these fish are very visually acute. They can actually tell individuals apart by their masking patterns, so we think that's pretty important. So we'll have the transcriptome, and now we can actually compare gene sets and see if gene sets are conserved by certain behavioral suites or size. And then what's really fun is we can make a priori predictions about expression levels in biosynthetic enzymes or nuclear receptors. Do females increase their androgen receptor to be more sensitive to that circulating androgen, whereas males may already have high levels? So these are types of questions we can ask along the pathway using the transcriptome already in hand. So with that, I'd like to thank you all for listening. I'd like to thank Susie Wren, who went off, who's been my postdoctoral mentor. She became a program officer for 2 years at NSF and handed me the keys to the lab. Very bold decision on her part. And it's been great to work with all these undergrads and produce this research. Right here are the undergrads I've had this summer, and we've basically done everything I just showed you, including going after brain punches and another species of Geliotochromis so we can have a more robust data set. So hopefully look for the transcriptomics on the transcriptist to come out, as well as a whole new thing on another type of Julie that does slightly different in its behavioral stuff. So with that, I'll take any questions.
curious about those like 4 pairs that like really immediately like each other.
Yeah.
Is there anything about sort of like that partner compatibility that might influence the hormones that you're measuring?
So I would love to know that, but it's just the sample size is so small that I really couldn't pull that out. What I did to confirm that progesterone is I took the non-focal fish and measured their levels too. So their partners that we gave them had higher than expected progesterone levels. So that's why I know that's playing a role. But no, I have not looked at corresponding hormone levels, right? Because I did not sample prior to introducing the fish. I sampled at the end, so there's one flaw with the data. But it's because you put the fish in and you hope they work. And I'd like to tell you I could successfully pair them every time, but I would say we have a success rate of probably about 3 and 4. And they'll just straight up start murdering each other if they don't like the partner you give them. And what's even more interesting is they are more likely to fail as a partner if you go in the opposite direction. So when we did large female, small male, those are more likely to fail at partnering. So they really didn't want to be that way. Yeah.
Just a quick question. I wondered if you had maybe done some experiments working with non-acromis transvestitis, which has switched, so it's the female which is the dominant coloration pattern, and does a lot of the male roles.
Okay, so I was hinting at that other Julie, and that Julie is called Julidochromos marlieri, and they look almost like transcriptus, but they are female larger dominant roles. But just like the transcriptus, they're not heteromorphic. Like you literally have to look at their urogenital opening to see the different types of shape. But yeah, so we've done this exact same process because, right, transcriptus is doing cross-sexual transfer plastically, whereas marlieri has totally evolved it. And so we're wondering what that does. And anecdotally, I would tell you the marlieri pairs are less likely to succeed when we do male larger, female smaller. So it's kind of this weird stuff going on. So yeah, the next question is with marlieri. But what was the name of that other?
Non-acromis transvestitis.
Okay, well, yeah. Yeah. All right. Yeah, sure. Thank you.
Thank you for coming to this talk, even if it's late on a Sunday, on a nice Sunday afternoon. So my name is Pierre-Olivier Montiglio. I'm a prof at University of Quebec at Montreal, a 5-minute walk from here, basically. And today I'm going to present some work on foraging specialization in predators, but in multiplayer video games, so in a virtual predator-prey system. And this is work that's been led by Maxime Fraser-Franco over there that's finishing his great thesis as we speak. He's trying to write it up and finish it. Okay. So the behavior that predators use when they forage, when they hunt for prey, or their hunting tactics, is really important if you want to predict the evolution of prey population. Or you can even have consequences for whole communities, right? You can think of trophic cascades, you can think of non-consumptive effects, indirect effects, and so on. So you have some predators that will use ambush hunting tactics. They will stay stationary in the environment or move at low speed, and they will strike when there's a prey that actually comes near. And at the other extreme of this continuum of hunting tactics, you have active pursuit. So these predators will actually just move a lot, move fast, and try to chase down their prey. And of course, you have a continuous variation between the two at the level of species, populations, and at the level of individuals as well. And one of the big modulators of this impact of predator behavior on communities and prey is the degree of foraging specialization, the range of hunting tactics that individuals can use when they hunt for prey. So you have specialists. They will actually use only one tactic or a narrow range of speeds or movement patterns. They will always ambush or always chase, for example. These are the specialists. And at the other extreme, you have individuals who are less specialized or generalists, and what they will do is actually, we think, they switch hunting modes. They use a wider range because they adapt, they adjust to the conditions, to the structure of the habitat, the behavior of their prey, their hunger level, and so on. So they have an increased plasticity, basically. But we still have a hard time to really pinpoint or figure out or explain why you have individual differences in foraging specialization within populations of predators. It's happening within the lifetime of the individual, so it's quite different than what we're trying to do with selection at the level of species. And I think if we want to make progress, what we really need to do is analyze how individuals maximize their hunting success as they gain experience within their life cycle. during these successive encounters that they have with potential prey as they fail, as they actually win and capture these different prey. And so it's pretty hard to do this empirically, obviously, because most predators are pretty elusive or too mobile. You need to track their behavior, hunting success, the type of prey that they encounter over time. And for all these reasons, and because there was a pandemic as well, in my lab we started using multiplayer video games. So we're using humans, or you, if you actually are a gamer, to answer those questions. And I think multiplayer video games are a great complement, perhaps, to the models and the simulations that we have in the literature on these questions, or several things about animal behavior, because they allow real-time ecological interactions. If you scratch past the themes of these games, it boils down to you'll have to forage to compete, you'll have to avoid predation or cooperate to defend resources. It doesn't become much more complex than that. And it happens in structured environments. And you have behavioral variability, right? You have people more or less good playing these games. And this behavioral variation is actually underlined by cognitive mechanisms and biases that we have in other animals. We're as dumb as other predators. And it's structured by the same processes: flexibility, plasticity, consistent individual differences, and so on. So you could see us revisiting basically all of our theories about animal behavior, but injecting all the stuff we conveniently ignored until now, if I'm skipping the nuances. So the game we use is called Dead by Daylight. So it's a scary game. It's modeled after the horror movies in the 80s and 90s. So you're meant to play it in the dark. That's why you don't see much with the light here. And in this game, you have 5 players interacting. So 4 of the poor players, have to locate resources, distribution on patches in the environment. And they have to accumulate enough resources to actually escape the environment. And they have to do that while avoiding being consumed in gruesome ways or horrible ways by the 5th player, which is the predator. Okay, so the predator moves around. He has to chase down the prey or ambush them, capture them, handle them. There's a handling site. And then he needs to consume them. So I'm not showing the whole thing. You can play the game if you're interested and bold enough. So it's a good game to actually revisit these predator-prey interactions in simplified settings, basically. And it all happens in this 3D structured environment. You have different maps, different types of habitats with varying levels of complexity, different types of distribution of resources. And it's procedurally generated, so you have replicate habitat structures, if you want. Maxime has been analyzing the behavior of players for a while now. He's finishing his thesis. You can read about these other results in our paper published 2 years ago. And what he found is that predators in this game actually use hunting tactics that fall along this ambush active pursuit continuum, just like animals in nature, right? The big continuum that were studied more in the 90s and the 2000s. Individuals differ consistently in the proportion of ambush and active pursuit that they use, but there's also some flexibility or some plasticity. Predators actually adjust to some extent their hunting tactics in response to prey space use, to prey activity level, and to their foraging behavior in general. Okay, so you can read all about this. And just a little bit of variability that you have in prey behavior among the 4 prey from game to game is enough to generate all the types of selection you can think of or you can imagine on this predator hunting tactics: correlational selection, contextual selection, and so on. If you're interested in the prey, the life of the prey, so Francesca Santostefano, who's right here, can actually tell you more about it. She documented the whole selection regime as well as part of coming from the social interactions among the prey. So our question is, how does experience shape hunting tactic specialization in this game? So what we did is that we, in partnership with the company, Behavior Interactive, which is a cool name, right? It's been editing the game in Montreal since 2016. We assembled a data set that comprises 243 predators, and we tracked these predators. We monitored all of their game from their very first game for a period of 6 months. So it gives around 500 matches for most of these players, right? So it's actually quite a lot of time in front of a computer game. I wish I had that time. And for each of these games, we got information on hunting success, number of prey that they actually consume during each game. It ranges from 0 to 4. The phenotype of the prey, all sorts of behavior, but we mostly focus on speed of movement as the main summary variable to summarize behavior of prey. The average speed of the predator, our best measure, or best proxy of its hunting tactic. And we used double hierarchical models in a Bayesian framework to analyze all of this stuff. And what we did is that we basically modeled the intra-individual variability, so the range of foraging tactics that you use and how it changes with experience over time, over the successive games that you play. First thing, you see that it takes quite a while to actually learn how to hunt, even for human beings in front of a computer, right? So here you have the hunting success of predators on the y-axis as a function of the number of games that they've played since the beginning in the game. And you can see that it takes a good 300 matches before hunting success stabilizes, okay? So there is some expertise emerging. Individuals actually maximize their hunting success over time as a function of what they've encountered in the game. Now the question is, do they do this by specializing, narrowing the range of foraging tactics that they use, or do they actually generalize or become more plastic and they actually increase the range of foraging tactics that they use? So here's a sample of our results for Predator 480,837, because everything is anonymized. That's a nice little name. You have the distribution of foraging tactics when it was a novice and didn't know much about the game for the first 100 games or so. And then you have the same distribution for that same player when he was an expert after 400 games, from the 400 to 500 games. That's in orange. So you can see that in the beginning, it's actually quite wide. The distribution of speed is actually relatively wide. This individual is using a wider range of foraging tactics. But as we actually get towards the expert phase, this player is actually specialized to some extent. It's actually decreased the range of foraging mode that it had. If you zoom out and add 4 more players as an example, you can see that individuals differ in their level of specialization with experience. So some players specialize a lot, right? Like these 2 players at the bottom panel, they actually really narrow the range of speeds that they use over time in these games. They specialize on one foraging mode, basically, and they use it really well. But you have other players that don't specialize much. They shrink only a tiny bit the distributions of their foraging mode. And if you zoom out still, you look at all this variability, it looks like some players even decrease their specialization. They increase the range of foraging modes that they use over time as they learn to maximize their hunting success. So they learn perhaps to switch better, right? Or to read environmental cues and to change how they play better in this case. So I give you 4 examples. You can see that some of them actually come to become so generalist that they will use the whole range of speeds that's available to them in the game. So it looks like just everybody seems to be able to increase its hunting success, either by specializing, either by becoming less specialized, by generalizing. So we're trying to figure out what predicts whether you specialize or not. And the main predictor is basically the variability, just the random variability in prey behavior that you encounter during your development as a predator in the game. So because you have individual differences in anti-predator behavior and foraging behavior among the prey, again, this is something that Francesca can tell you more about, those random differences create differences in the experience of these predators, and it allows the coexistence of specialists to some extent and generalists in the same population. So another less perhaps exciting way to say this, but more interesting if you're a theoretician, is that the null expectation is that you should have individual differences in foraging specialization within populations anytime you have individual differences in prey behavior, which is all the time, right? So the question is, why do we not have specialization in some population, or we don't have this variability in some population? That's becoming the more interesting question now, I think. So on this graph, you have each dot is a predator, and you can see that you have the variability in the prey that they encountered. If you're on the right, you encounter more variable prey. On the left, you encounter prey that are always moving at the same speed, whatever the average speed it is. And on the y-axis, you have the degree of specialization when these predators are becoming experts. So generalists are up, and the specialists are near the 0 on this axis. And you can see that there's a covariance, a correlation between the two that's actually quite strong, given that we're actually abstracting everything over variances. And we found that even after correcting for the behavior of prey, we still have a slight advantage in hunting success of specialists over generalists. So it seems that even after you account for all these differences among environments, specializing is still the way to go if you want to increase your hunting success in this game. So I guess the question now becomes, what are the costs of becoming more plastic and switching hunting tactics that really give this advantage to the specialist? So I'll leave you with this result. It's been 13 minutes, and I'd like to thank you for your attention. I'd like to thank Behavior Interactive for helping us with the data, funding, and that's it. Thank you. I should actually keep this, right? Do we have time for questions? We have 2 minutes for questions. Yes.
Does a predator ever have a chance to be prey, or does a predator only ever play the role of predator?
They can be prey if they want. In this data set, I think we focused only on the predator side. Yeah.
Okay.
But they could switch. Right? You log it to the game, you can play predator or prey.
You think maybe if a predator had the prey perspective, that might change their strategy?
It could. I think Maxim would be better person to answer that, but I think in preliminary analysis, we looked at this. Number of times you played as prey as a covariate didn't seem to have much of an effect. Yeah. Yes?
Does this game have like a ranking system where you got progressively more
Yeah.
challenging opponents, and then I guess, right, how would you account for maybe the higher level players, you know, around 400 and 500, like, okay, specialist works, I'm going to have fun trying tactics that I know are disadvantageous.
Yeah. No, it's totally possible. You're right. They should explore, right?
Right.
We weren't sure if it would be this or the opposite. Because you should explore when you're actually new to the game.
Right.
You do lots of stuff. Some of it works, some of it doesn't.
I'm thinking not from a word perspective, but they know a tactic is intentionally harder,
Yeah.
but they do it anyway just to challenge themselves.
Yeah, it's definitely possible, right? So I think this is one of the limitations. We cannot really look at the motivation
Exactly.
behind what they're doing, but it still has the same consequences, I guess. So, but we're correcting also for ranks in this case. So everything we do here is corrected for the fact that you play against harder prey as you become more experienced, which in this case translates into prey increasing slightly their speed of movement. But yeah, tried to account for that. Yes, there was a...
If we are translating this to, let's say, a real world predation scenario, would you consider you have like ambush predators and you have posing predators, right? So would you put specialization in the courses or the ambush?
That's a good question. So there is a correlation. I mean, it's not completely independent because speed is positive at all time, right? So if you're ambush, you have to be a bit more specialized than if you're in the middle, because there's more room for marks. Sorry?
Ambush can be also highly variable, and that's because the fact that they are predominantly at zero speed, and then when the prey comes in, the
Yep.
speed kind of varies.
I mean, they don't get involved in chases. So it's the average speed that we're using. So this should actually be removed from our analysis. But you're right. So I think time's up. Thank you for your attention.
Okay, hi everyone, I'm Tina Barbasch. I'm a postdoc at the Institute for Genomic Biology at the University of Illinois. Really excited to be here to talk about some of my research on flexibility in behavior and gene regulation in three-spine sticklebacks. So reproduction is really fundamental to fitness, but reproduction is also a risky activity. And in the case of predation risk, reproduction can even be deadly. And in many animals, males are brightly colored, making them attractive to females. Likewise, attracting females often involves conspicuous courtship calls or courtship displays. But these same traits that make animals attractive to mates might also raise their risk of predation. So bright coloration might make you more visible to predators. And these conspicuous displays can attract predators in addition to mates. So while there are many examples of how this trade-off between survival and reproduction is resolved over evolutionary time, for example, by shaping these sexual signals or favoring alternative reproductive behaviors that lower the risk of predation, the availability of mates and the risk of predation also varies through space and time. So what this means is that these decisions over how to respond to predation risk also play out in real time. And many animals are capable of flexibly adjusting their reproductive behaviors depending on their assessment of predation risk, as well as their expected current and future reproductive opportunities. However, the fundamental mechanistic underpinnings of behavioral flexibility are not well understood. And identifying these mechanisms and how they relate to decision making is really critical to our understanding of how animals manage these multiple competing demands, as well as how such behavioral flexibility can evolve. So my main question then is, how do animals balance this opportunity to mate against the risk of predation? And what I predict is that the need to manage these competing demands might correspond to flexibility in behavior, as well as gene network structure. So brain gene expression profiles often show really remarkable concordance with behavioral responses to diverse environmental stimuli. But gene expression profiles, in turn, are coordinated by these complex regulatory interactions between transcription factors, TFs, and their targets, which form these gene regulatory networks. And at the network level, behavioral flexibility might correspond to flexibility in network structure, gene network structure. And we can use dynamical systems theory to find these features that contribute to flexibility. So for example, in panel C down here on the bottom right, you see examples of two different GRN structures. So whether you have many of these TF-TF interactions or fewer, and it can be kind of visualized on this landscape where you have either deep valleys or shallow valleys representing the stability of these cellular states, which then correspond to behavioral states. So if we have fewer regulatory interactions, we get these shallow valleys with greater kind of ease of transition between states, so we get greater flexibility, but this comes at the expense of stability. So what I predict is that when animals are faced with these multiple competing demands, these trade-offs, flexibility might be favored over stability compared to when they're only managing one thing at a time. So to test this hypothesis, I'm using the three-spine stickleback, and stickleback might be familiar to many of you. They're really famous among evolutionary biologists for the repeated colonization of these marine forms into various freshwater habitats, but they also attracted the attention of early ethologists, including Nobel laureate Nico Tinbergen, because they have really diverse behavioral repertoires and really dynamic reproductive and social behaviors. And so because stickleback can mean so many different things, I think it's really important to mention that I'm focused here on one particular population of stickleback, a limnetic form found in Alaska. And stickleback males frequently face trade-offs between courtship and predator avoidance. So during the breeding season, males develop this bright blue coloration, these bright blue eyes and red throats, and then they court females with these conspicuous zigzag displays. So I think I can play this video. So here you see an example. There's that gravid female in the upper right corner, and these male, brightly colored males come around and court with these zigzag displays. But these same traits that make them attractive to females, so this bright coloration and conspicuous behavior, might also make them more vulnerable to predation, for example, by this rainbow trout, which is a common and voracious predator of adult sticklebacks. So using the stickleback system, I have three main objectives. First, I want to investigate behavioral decision-making by individuals faced with the trade-off between courtship and predator avoidance. Second, I want to identify the molecular mechanisms associated with decision-making. And then finally, I want to test this hypothesis that behavioral flexibility in response to these trade-offs corresponds to flexibility in gene regulatory network structure. So first, I'll investigate behavioral decision-making. And I presented male sticklebacks at their nest with 4 different treatments. And you can visualize this in a 2x2 design where courtship opportunity or risk of predation was either present or absent. So we have a control where they were presented with 2 empty cages, so both absent. A courtship opportunity where we put a live gravid female in one of the cages and an empty cage. The predation risk treatment, where we use this realistic model of a rainbow trout in one of the cages, in an empty cage. And then this trade-off, so where they have to manage both together. We have a live gravid female in one cage and a model trout in the other. And to show you what these fish had to go through, we have our model trout in the front here, and we would simulate movement of the predator. And in the back, you see the gravid female, and this male has to kind of divide his time between these two competing stimuli. And what we found was that males interacted less with the females under the risk of predation. So on the x-axis, we have our 4 different treatments, and on the y-axis, we have the log-transformed pecs at the female cage. So females in quote, because the control, it was toward an empty cage, as well as the predation risk treatment. And this was a measure of how much the males interacted with the females. And if we compare the courtship opportunity to the trade-off treatment, you see that males interacted significantly less with the female when he was under the risk of predation. On the other hand, we found that males retreated at a similar rate even when there was also a courtship opportunity present. So again, our 4 treatments, and this time we're looking at the number of retreats, so these rapid backward swims away from the predator. And if you compare the predation risk treatment and the trade-off treatment, you see that males are retreating at a similar rate, even when there's the opportunity to court. So to sum up, when faced with the risk of predation, stickleback males prioritized predator avoidance over courtship, reflecting the shift toward prioritizing future rather than current reproductive opportunities. Next, I wanted to identify the molecular mechanisms associated with decision-making. So I took brains from these males and I separated them into these 2 gross brain regions, the telencephalon and the diencephalon, so we could begin to gain insight into whether there are brain region-specific mechanisms associated with decision-making. And then I looked at differentially expressed genes in each experimental treatment relative to the control. And what we found was that the need to manage these trade-offs between courtship and predation risk elicited a really distinctive response in the brain. So these Venn diagrams show the sets of differentially expressed genes in our experimental treatments relative to the control. And while there is some overlap between the treatments, what I want to point out is that there were these sets of genes differentially expressed only in the trade-off treatment and not in either of the other treatments alone. And comparing across our 2 gross brain regions, the telencephalon and the diencephalon, again, we find a distinctive response in each brain region. So these are the trade-off genes from the telencephalon and those trade-off genes from the diencephalon. And this, to me, really highlights the importance of good spatial resolution to understand the link between brain and behavior. So to sum up, the need to manage courtship and predation risk elicited dynamic changes in gene expression. And these changes were more than just an additive response to courtship and predation risk, suggesting that there's something unique about needing to manage these competing demands at the same time, something special about the need to manage trade-offs. So I wanted to test whether this special thing is behavioral flexibility. So does behavioral flexibility in response to trade-offs correspond to flexibility in GRN structure? So we inferred GRNs from our expression data using GenieTree. And GenieTree quantifies the strength of these regulatory interactions using random forest to predict the expression pattern of each gene in our data set based on the expression of a set of known transcription factors. And then for each treatment, we calculated what I called the TF-TF edge ratio, and that's just a measure of the stability of the network. So again, we can have many TF-TF regulatory interactions forming this more stable network structure with deep valleys and more difficulty to move between these behavioral states compared to fewer interactions, greater flexibility. So I tested the prediction that the transcriptomic landscape that's associated with the response to courtship opportunity or predation risk alone would be more stable, so would have these greater TF-TF regulatory interactions compared to that associated with males facing a trade-off. Okay. And I did find evidence for a more flexible GRN structure. So here on the x-axis, we have our 3 experimental treatments. And then on the y-axis is this TF-TF edge ratio. It's all you need to know is that lower values corresponded to greater flexibility and higher values corresponded to greater stability. And if we compare 3 treatments, we see that we found evidence for more flexible GRN structure in the trade-off treatment compared to the courtship and the predation risk treatments. This pattern didn't hold for the diencephalon, the other brain region, and I have some hypotheses for why these 2 regions might differ, but I think it really highlights the need for future studies to explore whether these patterns hold in other contexts, particularly in other contexts where behavioral flexibility is favored. So flexibility in behavior did correspond to flexibility in GRN structure in the telencephalon, and this result kind of establishes a framework for linking that gene regulatory network architecture to behavior, opening the way for future studies. And so why do we care about this? Well, investigating behavioral flexibility can help us understand and better predict how animals will respond to ecologically relevant trade-offs. Identifying the mechanistic underpinnings opens the way for studies into how selection on flexibility can shape genomic architecture. And then finally, expanding this framework to other organisms and trade-offs can provide insight into the evolution of these decision-making mechanisms. Okay, and thank you to all of the people that helped with this, and thank you all for coming.
Thank you for hanging on to the bitter end. I'll try not to make it too bitter for you. Title of my talk is Quantitative Genetics of Personality and Plasticity in an Emerging Model Decapod. And I'll just start by acknowledging my 2 co-authors, Francesca, who's in the room over there and asked to be mentioned. Make of that what you will. But much more importantly, actually, is Rosie, who's not here. And the work I'm presenting today is actually work from Rosie's MSc thesis, which she had approved with no corrections last week. So well done for her. Okay, still not quite there though. Oh, maybe we move on to here. Yeah, okay, all right. Okay, so we're going to do it this way. Right, so this is a talk about personality. If you have been living under a rock for the last 2 years, you may not, 2 years, 2 decades, you may not have encountered this phenomenon, but assuming you have, because you're interested in behavior, then to sort of bypass all the confusion and arguments about semantics, when I say personality, I just mean nothing more and nothing less than repeatable behavioral differences among individuals that are sort of consistent across either time and or context. And we can think of those differences quite often as being kind of aligned along some kind of latent variables or axes or personality traits that we can give names to, like boldness or sociability or aggressiveness. And some people think that's a good idea, and some people think that's anthropomorphizing too much. I don't want to get into any of that. It's just variation among individuals that we're talking about. So there's been a huge explosion in research in this area and in using this kind of approach. It's still a little bit controversial in some circles. So some people in behavioral ecology would still argue, I think, that personality hasn't really provided any new novel insights into behavioral evolution. And in fact, some people would even suggest that it hasn't really generated any new testable hypotheses. If anyone in the room feels that way, that's okay. We can still be friends, but you're wrong. So for me, at least, I think personality research is useful for at least a couple of reasons. And the first one is that it forces us to think quite carefully about levels of variation and to be quite precise about the level at which we target our hypotheses. I think that's important because in evolutionary ecology, at least, we're sometimes a bit sloppy with this, kind of fudging across levels quite a lot. But actually, when you think about it, if you're interested in selection, most of the time you really are interested in differences among individuals. If you're interested in phenotypic plasticity or perhaps resource allocation trade-offs, then strictly speaking, that's a within individual or even within genotype process, right? So we sort of think about these levels, we can target them more appropriately. And the other thing is sort of even more simple, I think. We're all evolutionary biologists, and I think all evolutionary biologists probably believe that questions about variation are more interesting than questions about the mean, right? So by focusing on variation within populations, we are actually focusing on that raw material for microevolution, right? So there's all kinds of interesting questions that we can ask about that. And in my group, among other things, we've been thinking about these sorts of questions, mostly using small fishes as a model. So today I'm going to talk about personality in cherry shrimp. It's a slightly odd decision because I know almost nothing about this animal. But in my defense, not very many people do know very much about it. But I've started to think that it could be a very useful lab model for evolutionary behavioural genetics studies. So what do we know about it? We know that it's native to Taiwan, but actually it's much more widely found than that because it's gone into the global pet trade, and so everybody's keeping it all over the world. The wild type is a sort of little brown shrimpy thing like that, but it's been selected into all kinds of different colours. So the most common one you'll find in the pet trade is red. Now, as potential model species go, I'm going to be honest with you, it's got some downsides. The phylogeny is a little bit murky. There's a few things published, and I think it's probably resolved, but it is a bit murky. The ecology, wild ecology, really poorly described, at least in the English language literature. If anyone knows people in Taiwan,